So if you remember from cell theory, from the first things we talked about in class, you'll recall that all cells come from previous cells. So now I have a problem. How do we get this 2-meter long strand of information to be perfectly copied to the next generation? That's what this lesson is about. How do cells manage this huge feat of duplicating all their information that guides everything they do? This is it. This is how they do it. This is a computer simulation of how DNA is replicated. You'll see on the left there's one strand incoming, and on the top and the bottom 2 other strands are being copied off of it. Remember we talked about anti-parallel strands? This is what we mean. Notice the bottom strand is just continuous. It runs in one direction. The other strand is going in kind of loops. It runs anti-parallel, and DNA can only be synthesized in one direction. We won't get into why. It's some really gnarly biochemistry having to do with the structure of DNA. But this is the basic. So let's break this down, because this graphic, as cool it is to watch, doesn't tell us too much. So let's break it down a little bit. There. A little easier to see. So the blue is the parent strand, the old strand. And what it does is DNA kind of unzips, it unzips, and new nucleotides are brought in to match up. Remember, A with T, G with C. This is how the information is conserved. The A and T's, G's, and C's from the parent strand here. So let's just say this is an A. I mean, this will be a T over here. This is a G. So it'll end up being a C, right? And it just kind of goes on and on like this. So from one strand, you can duplicate the other strand because of a specific base pairing. Right, so let me erase my marking before I change slides. So this is the basic idea. So DNA unzips. This is all done by protein enzymes. DNA doesn't do this by itself. Excuse me. And creates 2 identical daughter strands. Hopefully identical. Sometimes there are issues, but we're not going to worry about exceptions for now. Right? Point being, every daughter strand of DNA has half of it's from the original, from its parent, and another half is new. This is a little more involved in it. So we have on the right side now the parent strand. You can see all these little strange shapes. Those are all different protein enzymes. So the first one kind of unwinds it, and then the helicase, this means it unzips it. It takes it out of the helix shape, and you can see on the top, sorry, you don't even know all these names of proteins. What we're showing is just how proteins are involved. And sorry, on the top, you see kind of lagging on the loopy part from the last slide. That's what this is, because it can only synthesize DNA in one direction. We're not going to worry about that. Why that is, just that it is. So we need, these are called Ozaki fragments, which Ozaki is the scientist who first discovered this. So DNA on one side is synthesized in chunks, and then it's bound back together by another enzyme. It continues on its way. This is really important just biochemically of how does the information conserved given the complexity of this molecule. So DNA is replicated, it unzips, it unspirals, and it splits. And then with specific base pairing, creates 2 new strands. So summary, again, the parent strand unzips. This is how DNA replicates. The parent strand unzips. Complementary base pairs join up to the exposed parent strands. Phosphate sugar backbone is built alongside these base pairs. And then the strands twist back into their own double helices. Clear? I hope so. Please see me in class. We'll do some work together to untangle this situation some more.